Welcome to another screencast where I'll be discussing the endomembrane system and its role in post-translational modifications of the polypeptide. So by the end of this you should be able to describe the components of the endomembrane system and then describe and explain the role of each of the following in post-translational modifications. So we'll be looking at what the rough ER is doing, what the Golgi apparatus is doing, and then what vesicles and lysosomes are doing in the endomembrane system. So let's get started. So we've looked at, in previous screencasts here, the expression of DNA, of genes here in DNA. And so this is showing you, of course, the transcription, post-transcriptional modifications here, the mRNA, the mature mRNA leaving the nucleus here, and then being translated here into a functional or a polypeptide. Now this polypeptide may or may not be functional, and most often it is not functional, just the way that it is assembled. So there are post-translational modifications that are going to happen, and so this is going to be modified, right? And where does that happen? Well, that depends on the protein, but in general the endomembrane system is going to be involved in this. So if you remember from previous screencasts, we talked about how membranes separate solutions or separate fluids here. So the endomembrane system, the endo in endomembrane is within, right? So the membrane's within the cell. So we're talking strictly eukaryotic here. The nuclear envelope is part of that endomembrane system. The rough endoplasmic reticulum is part of that endomembrane system. The smooth endoplasmic reticulum is part of that endomembrane system. Sorry, still getting used to using my new pen here. The smooth endoplasmic reticulum, as well as the Golgi apparatus. And then you see several types of vesicles here that butted off of that Golgi apparatus. And then lysosomes also can be considered part of that endomembrane system. So what's not considered part of the endomembrane system are mitochondria, centrioles. Well, they're not membrane bound anyway. And if this was a plant cell, we would see chloroplasts here. They are not part of the endomembrane system. So what's happening here? Well, let's take a look at the nucleus first. We had talked about the nucleus in a previous screencast. So it's a double membrane system here, but I want to point out this part of the cartoon right here and how this outer membrane of the nucleus is actually continuous with the membrane of the endoplasmic reticulum. So this endoplasmic reticulum is membrane, right? And that means that it's separating the cytoplasm from a lumen, right? So this would be the lumen, or the space lumen inside of that endoplasmic reticulum, and out here would be the cytoplasm. And so what's inside the lumen here are going to be a whole host of enzymes that are going to be modifying proteins. Okay. Now, if you're comparing the smooth endoplasmic reticulum with the rough endoplasmic reticulum, so over here, smooth endoplasmic reticulum, and over here is the rough. So the smooth endoplasmic reticulum is going to contain enzymes which do these things, right? Synthesize lipids, help metabolize carbohydrates, detoxify drugs and poisons, and regulate calcium ions in muscle cells. We're primarily going to be focusing over here on the rough endoplasmic reticulum and how this has enzymes that are going to produce secretory proteins, and also then new membrane. New membrane for the cell, new membrane for the ER, new membrane for the Golgi, new membrane for the nucleus, right, etc. Right, so by secretory protein, I mean a protein that's going to be secreted out of the cell, and we're going to use a specific example of insulin to demonstrate this later on. So what we're seeing here then is the cytoplasm, and we're going to look at the rough endoplasmic reticulum. So this would be the lumen of the rough endoplasmic reticulum. And so here's the membrane that is separating the cytoplasm from the lumen. They also sometimes refer to this as the cisternal space. A cistern is a holding tank, right? But in that membrane, there are translocation complexes. These are, you can think of these as pores or as receptors. Again, these are internal receptors here. And so what's happening is you get the ribosome translating this piece of mRNA right up here. And the first bits of the polypeptide that come off there are known as a signal peptide. They're going to bind with this SRP, this signal recognition particle. Well, when this binds here, you get this binding, and that SRP is going to guide that whole ribosome with this mRNA and this elongating polypeptide right to this translocation complex on the endoplasmic reticulum. And so as that docks and binds right here, the pore opens up, and the docking of the binding then releases the SRP, pardon me, and the continually growing polypeptide is then made and dumped into the lumen of the ER. Upon completion of when you reach the stop codon, when this ribosome reaches the stop codon up here, the whole complex then dissociates and goes off to be reused again. And now you have this protein, but the point here is that this is probably not yet functional. Right? But this protein is destined for to be a protein in a membrane or to be secreted out of the cell. Okay, so let's continue to follow the path of this protein. The next place that's going to go, what's going to happen is this protein that's here, this part of the membrane is actually going to bud off of the endoplasmic reticulum, and it will then fuse with the Golgi apparatus, right? And so you have two sides of this Golgi. This is the cis side, and then here's the trans side. All right, so the cis is in and the trans is out. And so here's this transport vesicle from the ER that I talked about that is budded off, so our little polypeptide thing is in there. This is going to then fuse with this part of the membrane of this Golgi apparatus, and this is a multi-membrane thing here. And there's an animation on another slide that shows exactly this happening in real time. But suffice it to say that the lumen of this Golgi here also has enzymes that are going to be modifying this protein, this polypeptide that was coming from the ER. Right? And so as the protein makes its way through this membranous complex, it's going to come out here and will either then be destined to be secreted from the cell or become part of the membrane. So here's another image of this, right? So again, here's our ER. There's our vesicles containing enzymes and containing our polypeptide bits in there. You see these vesicles fusing with the Golgi, and then so this would be the cis side of the Golgi. This would be the trans side of the Golgi, and then these are vesicles that have budded off. You notice that how this now is modified here. It looked different than when it went in. And then you have exocytosis, where those vesicles fuse with the cell membrane, and the contents then are dumped to the outside of the cell, and you get this secretory vessel here. So there are lots of animations here that are also on Blackboard to take a look at to show this whole process. Right? So, as an example, you've heard of the enzyme, or the hormone insulin, and what we're seeing here is this is the insulin molecule. There's an A chain, there's a B chain that are connected together by disulfide linkages. Right? So this insulin molecule is, of course, these are all amino acids, and so the primary structure would be these things just strung together, but the functional insulin molecule is not just one, it's not a single line like this. You have to take that single line and then it has to be cut and then put together. So you cut it and you get two pieces, and then those two pieces have to be aligned like this, and one folds over like this, and you got to bond those two pieces together. So this is then what that functional insulin looks like. So over here the schematic is, here's my ribosome docking to that translocating protein. This is the membrane of the ER right here. So this would be the lumen of the ER right over here, this white space, and you see that all of these letters represent the different amino acids of that insulin molecule. So this comes into the lumen of the ER. This represents an enzyme that's going to then cleave this part here. These are other ones that have already been cleaved and are now going to enter a vesicle and leave the ER and into the Golgi, and as it works its way, it being the immature insulin, as it works its way through the Golgi, you get other enzymes here and here and here that are going to be doing essentially what I was describing up here, right? Cutting this, binding these two things together, cleaving it, so that then by the end you have this last enzyme here, this part, carboxypeptidase E, that produces the mature insulin, and then that mature insulin right here will be destined to be secreted out of the cell. Okay, and of course this is happening in cells of the pancreas, and specifically beta cells of the pancreas. This is where that's happening. Okay, so just a specific example of how a protein might be modified from its original polypeptide string right here to then this functional mature molecule that's actually going to then leave the cell and provide some sort of function. Related to those vesicles are lysosomes. So when these lysosomes contain enzymes that will be degrading cell parts that are being recycled, or in this case, so we see this happening over here. So here's a lysosome. This is going to then fuse, and you get autophagy. So these are old cell parts here. This could also represent faulty proteins, right? Faulty cell parts there. When you get substances coming into the cell, they are filled in with, or they are endocytosed into a vacuole, and those vacuoles then will fuse with lysosomes, and so then these little dots here are digestive enzymes or hydrolytic enzymes that are going to then break down that molecule, that particle, etc. Okay, just related to the vesicles there. So hopefully now you'll be able to describe and explain the components of the endomembrane system, and then talk a little bit about the role of each of these, the rough ER, the Golgi, and the vesicles. And the example we used was insulin to show all the characteristics of the endomembrane system and post-translational modification. Okay, thanks for listening. Bring your questions to class.